Good day. Today I will discuss chapter 2 of our book, which is on the next biomolecule, lipids. So here is an outline. I suppose you are familiar with these things now. So these are the things that you will be able to learn and understand um, during the duration of my discussions for chapter 2. So to begin with, we have here the definition for the word or the term lipids. So lipids are organic compounds found in living organisms that, take note of this one, insoluble or only slightly soluble in water, but soluble in nonpolar organic solvents. So unlike other biomolecules, like say for example carbs or proteins, these lipids do not have a common structural feature that serves as the basis for defining such compounds. Okay, so unlike other biomolecules now, like say for example carbs, wherein classification is easy because you just have to look at their common structural features, that is not the same, or that is not true with lipids. However, we classify lipids using two methods, and these methods are based first on their biochemical function, and the second method is based on saponification. So moving on, we have here uh, an, an illustration of the diversity among the different types of lipids. So we can see in the figure right here the structural diversity that is associated with lipid molecules. So some of these lipids are esters, some are amides, and some are alcohols. Now, if we take a look at all these lipids, example lipids that we have here, we can see right here that they do not have a common feature which, like at first glance, you would be able to classify them immediately as lipids. However, the common thread that ties all of the compounds that can be seen in this figure is their solubility rather than the structure. Okay, so if we take a look at the illustrations of the different example lipids as shown in this figure, we can see that their structures indeed are very, very diverse. So the common thread that would tell us that these group of compounds are indeed lipids is because they possess the same kind or type of solubility, and that is they are not soluble in water and other polar solvents. However, they are soluble in non-polar solvents. Okay, so more on the methods for classifying lipids. So again, we classify lipids based on the function. First, the function that they perform, and whether or not that lipid can be hydrolyzed. Hydrolyzed, of course, is the reaction with water under basic conditions. Now, if you recall our discussions on carbohydrates, hydrolysis of, say for example, a disaccharide would yield or would produce the two monosaccharide components that is composing that certain disaccharide. Now, in hydrolysis of lipids, later you will see that lipids are also composed of different components, and then when it undergoes hydrolysis, the result would also be the breakdown of these components into their singular state. Now, this process in lipids is what we call saponification. So later on, I mean more of saponification later. Okay, so let's try to discuss the classification of lipids based on biochemical function. So this is of course our first mode of classifying lipids. So based on the biochemical function that they perform, we classify lipids or we categorize them into five. And these five categories are energy storage lipids, membrane lipids, emulsification lipids, messenger lipids, and protective coating lipids. Now, from their very names, we can already see what kinds of functions they perform in our bodies. Now, of course, energy storage lipids are those lipids that stores energy. So example rather, of these kinds of lipids are the triacylglycerols. Now, the next category of lipids is the membrane lipids, which, as what the name implies, these are the lipids that can be found in the membranes of our cells, performing different functions. So, examples of lipids that are classified as membrane lipids include phospholipids, sphingoglycolipids, and cholesterol. The third category of lipids is the emulsification lipids, and these include our bile acids. Now, for the messenger lipids, these lipids, of course, responsible in sending or initiating messages between and among cells. Now, these includes steroid hormones and the eicosanoids. And then lastly, we have here protective coating lipids, which of course would provide coating the different organisms for the purpose of protection. Now, these are the biological waxes. So these are again the five categories which is under the classification of lipids based on their biochemical function. Now, if we talk about the classification of lipids based on saponification, we can divide lipids into two categories. Now, these are those that can be hydrolyzed, or the saponifiable lipids, and those that cannot be hydrolyzed, or the non-saponifiable lipids. So examples of lipids or lipids that fall under the category of saponifiable lipids are the triacylglycerols, the phospholipids, the sphingoglycolipids, the cholesterol, and the biological waxes. Now, the lipids that fall under the category non-saponifiable are those bile acids, the steroid hormones, and the eicosanoids.